right, let's get him started. All right, the next thing that we're talking about is allele-specific expression. All right, in this part, the take-home message I will tell you first is I don't believe that we have the way to do that at this stage, just because of the technology limitations. Okay, and so what is allele-specific expression? And this is the slides from Dr. Bimuro from Purdue University. So you can see that there's one gene, there's the exons, and there's the promoters, and there's happens to be one of the SNPs here. We can use as a marker, which is a tag SNP. And there's another one here, variation here that is in the promoter region. And somehow, if there's a transcription factor supposed to bind in this region, and that will control the expression levels. And if this star one is the right one, that means for this isoform, the expression level will be much higher than the other isoform. And because the transcription factor will have the ability to bind here because of a single nucleotide variation here, and it's not going to bind in this particular allele. Does that make sense? So we're talking about two alleles. One is from father, one is from the mother, and this is in the same cell. And so the conceptual way to do that is you can quantify that by counting the tag SNPs following the RNA sequencing data. So if you see there's so many reads contain this read, others contain this read, and you can see the allele imbalance. Actually, allele-specific expression is not only on the gene expression level. It can occur on any single stage. So you can see it can occur on the differential acetylation of histones. So you can see for different alleles, their histone distribution will be different, and that will cause allele imbalance. And the differential methylation of the DNA and allele-specific methylation, or RNA polymerase II occupancy difference, and differential concentration of nucleus mRNA, and differential concentration of cytoplasmic RNA, a different concentration of expressed proteins. So allele-specific expression is not a single event that only occurs on the gene expression level. It can occur on many different levels. The question here is, why do we want to study allele-specific expression? Okay, the gene expression is a complex trait that is influenced by cis-acting and the trans-acting genetic and epigenetic variations, and also the environmental factors. So just take an example. So when we look at, we do a study and we recruit human samples, and then you get a tissue, the people walk into the clinic, you get a tissue, and then you measure the gene expression. You are thinking, this is a disease, this is normal, and let's compare it. But it turns out to be not only it's caused by disease or normal, it also caused by what this person eat in the morning or what kind of medication that individual took, right? So all those different factors will contributing to the final expression level of the gene that you're measuring. And this is a typical setting of for the environmental factors, okay? Another more straightforward example of that is what is the genetic effect, what is the environmental effect. So some people give me an example. If your kids looks like you, that's a genetic effect. If your kids looks like your neighbor, let's hope that's the environmental effect. So, so the question here is whether there is something that we can rule out the environmental effect. Sometimes even the trans-acting effects, only focusing on the cis-acting effects. So in principle, the allele-specific approach can eliminate environmental and trans-acting factors. So two alleles serves as a within sample control. If we have a way to measure how much this allele express or how much this allele express, okay, if they are different, and most likely the difference will be caused by the cis-acting effects, okay, because they are living in the same cell, they ate the same breakfast in the morning, they took the same medication, right? Even the transcription factors and their concentrations or activity levels are the same. So the environmental effect and the transacting effect will be eliminated. And that is a perfect case for us to study the gene regulation. So even though some of the allele-specific expression, they do not represent severe phenotypes, but people still want to look at that, trying to figure out the mechanism that control the gene expression. And how popular is the allele-specific expression? Actually, the recent study suggested about 20% of the sites, heterozygous sites, show 1.5-fold difference between alleles. Okay? This is one allele, this is another allele. They show 1.5-fold differences, 20% of the sites. And 30% of sites show 1.2 differential in the allele differences. Okay? And again, as I mentioned, the phenotypical effect of this allele-specific expression sometimes is not really obvious because their differences, as you can see, are pretty subtle. But and also allele-specific expression is tissue-specific, and this reflects the transacting and the environmental effects. Okay, so this is some basic idea of allele-specific expression. Yes.
What's the margin of error when they say 1.5% margin?
No, this is based on the study that tells you about 20% of the sites have this 1.5-fold. You are asking for the confidence interval and things like that, right? You probably want to look at the original paper. I don't have the number in my head. But the point here is they are mild, but at the same time, it's not one or two things. And 20% or 30% of sites shows that. And in terms of the technology to study allele-specific expression, and there's two type of scenarios. And we will go to the next generation sequencing part, which I don't believe that we can do it at this stage. But before that, there's a polymorphism directed approach, which means that you already know there is a variation in this region, and you have the assay to directly target on that region. And so what the approach to use here is two. So the first one is you can focus on the primary transcripts, which you can see that not only based on the variations in the exon regions, but also in the intron regions. Okay? This is sometimes important, whether you are focusing on the primary transcript or mature transcript. And because the mature transcript differentiation can be caused by the splicing variation, things like that. And if you only want to see the promoter variation, how that will drive the gene expression levels, probably the primary transcript is a better approach to do. And another way to do that is use a more targeted approach, and it's based on the highly informative sites are captured. So we will talk about that. And the technology, the real technology that I think in terms of genome-wide scale to study allele-specific expression, and this is the only approach that I think will work. So this is used or designed in George Church's lab, and we know he's one of the pioneers for technology development. So what they did is called a padlock-seq assay. It's not directly from the RNA sequencing assay. So what they do actually is, I'm going to focus on this, and you can see that this is the variation that people want to identify. People already know there's a variation here. I want to see the variation in terms of allele-specific expression. And what they did here is they will design the probes. This is a probe, and which has both two ends that designed to target these variation flanking regions. Okay? So one is the 3' end, and one is the 5' end. And then they have this P1 and the P2 linkers, and those will be used in the sequencing part. And there's a restriction enzyme set here as well. So if you can imagine, once you put this probe into the cell, okay, what happens is the hybridization will happen in those regions. Okay, it will hybridize here, and you form this kind of structure. And after that, you do is you will do several steps. The first one is hybridization. And the second step is you want to do the polymerase extension, because we are still having this one nucleotide gap that we want to figure out what that is. And then you fill the gap with the polymerase, and then you use ligase to really connecting these together. Okay? And then you use restriction enzyme to cut this, and that's the part that you will get this final product, which what happens? They're vibrating? All right. So which is this product that has this variation inside it, but this is one connected product. Okay? And then you further do the sequencing on this sequencing rates. And by this way, you are targeting this variation event. Let me go through this again. And so you see this is a variation. Okay? You design the probe, and one end will target on this flanking region, and the other end will target on this flanking region. Okay? And these two different colors, they represent the linkers we will use in the sequencing reactions. And there's a restriction enzyme site in the middle. Okay? So this is kind of the probe you initially use. When you do the experiment, you first do the hybridization, meaning that this will hybridize to this. And the middle one will be a gap, because you do not know what that is at this stage. Is that me, or this thing is really evaporating? It's not my eye, right? It is evaporating, right? Okay, good. Yeah. I thought because I'm short of sleep. All right, good. So that's comforting. Thank you. And since there is a gap, and then we want to do the polymerase, and then to fill in this gap and use ligase to connect this together. And then this will be a circular thing. And then once you use restriction enzyme to cut this, and this will become what your final sequencing product. And then you sequence this using bioinformatics, you will be able to see that the number of reads contains this variation or the other variation. Yes.
Why can't you just do more?
I actually don't have a clear answer for that. That's right. That's right. Well, ligation could be, but it depends on the primers you are using. Okay, but I think Jake is right in terms of that. They will the way they do that is kind of they target over 10,000 variation sites in the genome, right. And by just changing the probes. And actually, the restriction, the ligation site is sequence specific, right? Yeah. But that's what we got. And then next step is sequence it. And this can be used to study allele-specific methylation as well. I remember I saw that one paper, but while I was preparing for this lecture, I didn't find that paper. But if you see it, let me know. So this is basically a very targeted type of technology to study the allele-specific expression. And that has a really high sensitivity and specificity. Okay, let's talk about the global approach using the RNA sequencing experiment. This is kind of a de novo way to look at that. So the principle is very simple, and you can use this is the RNA, and this is a variation, and there's a different transcripts. So you just count the numbers. This is the G, and this is the A, 7G versus 3A, and this can represent the allele imbalance. And you can use the binomial statistics to test allele imbalance or any other fancy approach. There's a lot of fancy approach available as well. But the challenge here is the first one is all the alignment algorithms are biased towards the reference allele. Okay? It's not surprising because we are, in a sense, that doing the alignment based on the reference against the reference allele. So there's a variation in the middle that already start to give some discount on this alignment. So all the algorithm is actually biased towards the reference allele. And the second one is it requires both RNA and DNA sequencing using the same sequencing platform. And this sometimes it can be really problematic. And because for most of the samples, we do not have the resource to do both DNA and RNA sequencing. And another hurdle here is hundreds of reads per base pair may be required for subtle variations. Subtle variation, we know it's only 20 to 30% of sites has 1.5 or less differential expression between the transcripts. So this is a figure that you probably cannot see from the back, but the point is X here is the number of informative rays, and this for this one it goes to 500 rays, and this Y is the power to detect allele imbalance, and this is 60%, and if the allele imbalance is 1.25, and you need 400 rays to cover that one nucleotide, which is sometimes is impossible. Okay, and let me blow this up again. Because I think this is one of the major justification I can, oh, here, I can do this. This is not the slides I'm looking for, where I am. All right. That one needs to be in the front. So you can see from this one is X is the number of informative rays per gene, and this goes to 500. And each line represents the differences in the allele imbalance. If the two alleles, their imbalance is 1.5, so one allele is kind of 1.5-fold more than the other one. And if you want to have about 80% of detection power, you need about 200 rays to cover on this particular site. So we know that at a current RNA sequencing experiment, very rare we can have that deep coverage for anything. And so I think at the current stage, this is a major hurdle to identify allele-specific expression. Does that make sense? Okay. And on top of that, you do need a DNA sequencing and RNA sequencing. You need both of them to figure out allele-specific expression. That is very resource-heavy. And next topic I want to go through quickly is the RNA editing, because this is another topic people constantly talk about using RNA sequencing experiment that you're capable of identifying RNA editing. And so what is RNA editing? We know that from DNA to RNA, this is transcription, this is translation, right? Where it's the hope that the protein sequence will be determined by the RNA sequencing. But sometimes the translation is not always faithful. We hope they are faithful, but sometimes it's not always faithful. So you can see this is the DNA. And the transcription, this GAC becomes the GAC, right? This is reverse strand. And if we do the right thing, the translation occurred here, and those tRNAs bring in the correct amino acid, and then put that into the final protein product. But when the RNA editing happened, this A somehow become a G, and this G will after the translation will become something else. It's a different amino acid sequence, and eventually kick into this protein product. So you will see that even the same RNA, initial messenger RNA, will become to different protein product. So this is RNA editing. And there's a little bit more biology about RNA editing. So the canonical RNA editing events is edited by deamination. So it's not like from A to C or A to T, from any two letters you can do this type of editing. It doesn't happen in that way. And it only occurs from A to I, well, I will eventually become G in the translation. So only A to G or C to U editing. Okay? This is a canonical editing. There's maybe some non-canonical ones, but this is the canonical one. From A to G or from C to U editing. And both of them, if you go through the initial structures of this, the purines permeatins, and you will notice there is a NH2 group here, and there's an NH2 group here. This is A, this is C. And once you get rid of this NH2, it becomes G, and this becomes U. So both of them actually is the animation process. And there are specific enzymes to catalyze both of these two events. This ADAR is responsible for A to G change, and this gene is responsible for C to U change. Actually, over 90% of our RNA editing is A to G difference. Okay, and again, a point I want to make here is this is different from any noise, transcription or translation noise. This is planned. Okay, so the cell made these enzymes to make this happen. So it does have, it's not a transcriptional noise. And there's other forms are also possible, but we don't know how frequent they are. Okay? And if we use next generation sequencing to identify RNA editing events, it's only possible when comparing DNA sequencing versus RNA sequencing. Okay? And not like our allele-specific expression. And if for certain regions it's really, really deeply sequenced, you probably know DNA sequencing is fine. But for the RNA sequencing, you have to have the DNA sequence accompanied with that. And this is the data from one of our collaborators. So you can see that at the bottom here is the DNA sequencing data, and top one is the RNA sequencing data. And this is clearly it's I want to see, this is a T to C conversion, okay? And we know it's not happening. It's not a canonical pathway, but it's on the reverse strand, it will be A to G conversion. So this tells you this transcript is on the reverse strand. But the point here is it's only possible when comparing DNA sequencing versus RNA sequencing. It requires a very deep sequencing to do that. Okay, and there's one paper published this year in Science, and they are using, I think this is from Vivian Chang's group at Philadelphia, and what the paper is widespread RNA and the DNA sequencing differences in human transcriptome. They did a fairly systematic analysis. What they did is they focused on 27 HapMap samples. Okay, the reason they did this is on the HapMap samples is for most of these HapMap samples, the DNA sequencing and variation data is already available. So we already know the DNA sequencing part. So they only need to do the RNA sequencing part. They use the B cells. Of course, that's what HapMap project provides. And the coverage is per sample is about 41 million reads. Okay, so but you can see from this type of statistics that if the DNA sequencing is available, you don't really need to get very, very deep. 41 million is enough. Some result summary, and they found that at least 10% of the reads covering a site differ from the DNA. That's a lot, 10% of the reads. And they found that over 10,000 exonic sites, and in total there's 28,000 events in 27 individuals. So there's really happened a lot in terms of RNA editing. Okay. So this one is that they require at least 10% of reads cover a site differ from DNA sequencing, and they call it one editing sites. Okay, and they identified over 10,000 those editing sites, and they found about 36% of expressed genes have RNA editing happened. So it's really widespread events. It's not individual event. And those editing sites are kind of recurrent as well. And this slide tells you the types of conversions that happened in the RNA editing events. And they did some statistics. They did from A to G, T to C. It's essentially every 2 letters, and they observed the differences in the RNA editing. This is very different from what I just talked about a few slides before. The canonical pathway can only edit from A to G or C to T. So they saw all those different events, and only these two are canonical editing events. And they also do the validation, major findings were validated. You use Sanger sequencing and even mass spec data. They did a validation. So it's kind of hard to argue with them about the results. And there are some people concluded this is a paradigm shifting paper. Okay, but is that really a paradigm shifting paper? And shortly after this paper was published, there's a Nature focus, and they have an article published called "Evidence of Altered RNA Stirs Debate." Okay, because not only their groups are working on RNA editing, some other groups they are as well, and they have a lot of resources as well. And I'm going to just label the 2 comments here. The first one here is some people suggested the sequence RNA makes systematic errors when sequencing RNA and DNA and RNA. He says that some of the chance mismatch occurred at the sites that are prone to systematic RNA sequencing errors. Okay? Sometimes the sequencing errors can cause those kind of artifacts. There's another piece of information says that another percentage source of error is multiple regions of similar DNA in the human genome can make it difficult to trace the origin of a short stretch of RNA to a specific DNA, meaning that this is implying that their alignment sometimes is not careful enough, and to conclude that. Actually, last week we came to a conference, and another group that working on the RNA editing was there as well, and they provided, not published, but very solid data suggested that most of the data that published in this paper are kind of artifacts. And, but the information I want to deliver here is RNA editing is a popular event. It happens a lot. But when we do the analysis, we really need to be very, very careful. The summary slides for this is RNA editing does exist. The canonical editing is A to G and C to T. Both DNA and RNA sequencing are required, and careful bioinformatics is needed to identify a credible RNA editing events. Okay? Any questions on RNA editing? I think the opposite side, and it's correct. That's my opinion on that. I think especially the conference I went to, and they in the talk they presented very, very solid evidence suggesting that once you do this filter, that filter, there's a couple of things that if you run more systematic checking on that, and actually the 2 events that stands out are the canonical ones. It's A to G or C to T, and the rest are really the noise. Yeah, I don't want to get into this debate, but I think I will lean to favor the opposite part. All right. We're still on time. Good. A couple of slides talk about non-coding RNAs, and this is small non-coding RNAs. So we talk about the ENCODE project. And ENCODE project concluded that 70 to 90% of genome is transcribed into RNA. And this is kind of change our view, right? And we used to think only 1 to 2% of genome will become RNA. And there's a lot of them are non-coding RNAs, which is functional RNA molecule that's not translated into protein, and is highly abundant and functionally important. And there's many different types of non-coding RNAs, okay? And specifically for 2 types of regulatory RNA, one is the microRNA, the other is long non-coding RNA. And I'm not going to talk about long non-coding RNA because the analysis itself is really not too much different from the protein coding genes. they are looking very much like protein coding genes. They have poly A, they do splicing, and they do all those kind of things. So the regular RNA sequencing experiment can take care of that. But microRNA is something pretty special at this case. So let's go through that. And the microRNA biogenesis, just a quick slide to review your biology. So we know from the DNA, the first thing happens will be the transcription. And the transcription will be used RNA polymerase 2 or 3 and to make the DNA into the RNA. At this stage, it's called the primary microRNA that can be 10,000 base pair long. It's very, very long. And then later, they form this hairpin loop, and they make this kind of modification, and then becomes the precursor microRNA. And this precursor microRNA is around 80 base pair, a little bit longer than that. And then after this stage, it will be exported into the cytoplasm and then further processed using dicer and JOSAR and several other enzymes. Eventually become this 22 base pair mature microRNA that will main trigger is to do the degradation on the genes. So those microRNAs, they're endogenous. That means the cells make it. It's not like siRNA we put into the cell and trying to kill a certain gene. And there's over 800, actually maybe 1,000 in human microRNA has been identified. And we know that more than 2/3 of human genes, they are microRNA targets. And the functions of these microRNAs, they are mRNA cleavage, translation repression, or mRNA deadenylation. Okay? Anything of this happens, it kills the gene, right? So that is the function. The question here is, I'm not trying to deliver how to do the microRNA bioinformatics. There's a lot of different aspects of that. Here, we want to focus on the detection. Okay? And using next generation sequencing. You cannot detect microRNA using regular RNA sequencing experiment. Because the first stage we have to fragment the RNA into smaller pieces, which microRNA is kind of already fragmented. And then we do the size selection for those RNA or cDNA molecules, the fragments. We want it to be about 200 base pair or from 100 to 300 base pair long. And then build the libraries based on that. So at that stage, the microRNAs has already been get rid of. They are too small, so they are not going to be in the RNA sequencing product. So you have to have some special assay to do that. And the first stage you'll do is so-called size selection. And then you'll want to focus on very small microRNAs, like this 19 to 24 nucleotides. And you only pick the ones that goes the fastest, right? They are smaller. And then what you do is you will ligate adapters on both ends. So you can see this end, you ligate one adapter. This end, you ligate another adapter. So both these two adapters will be ligated. And then you will further generate something, the product like this. You got a 5' adapter, and there's a couple of N here, and the microRNA is here, and a couple N here, and 3' adapter is here. Okay? And you may ask me what these N's are, right? And these N's is kind of, you can consider this kind of a library type of thing, because we know that ligase, those ligation enzymes, they do have sequence specificity sometimes. So you want to make it as comprehensive as possible. And actually, you can adjust this to a certain extent. And the point here is, in the final sequencing product, you have this 5' adapter, 3' adapter, and microRNA is sandwiched in the middle. Okay? And then at this stage, you can do PCR just based on the adapters, and then you can do the further sequencing part. And of course, we don't have to use 76 base pair sequences. We probably only use 35. That would be enough, because the real final product that we are interested in here is very short. It's only 22, less than 25 base pairs. But it comes to this point, there's a problem here, because if we sequence 35 base pair, and the sequencing actually starts from here, and because this part is in the sequencer already taking care of this part, so the sequencing from here, if we go into their 35 base pair, So we are not only sequencing this microRNA, but also a significant chunk of 3' adapters as well. So those are the things that we need to keep in mind. We are not only sequencing the microRNA, but also the 3' adapters. Okay? There's several analysis pipelines that has been published, and this is published in the NAR, I think, web server issue, and from Tsinghua University in Taiwan. And they call it DSAP, Deep Sequencing Small RNA Analysis Pipeline. So the strategy is simple. The first step they do is they will remove the adapters. So you can see those sequences, they are 35 base pairs, and we know that these are mature microRNAs, and that these are the 3' adapters. So you have to have some way to get rid of those 3' adapters. And this is a general strategy, right? It's pretty simple to do. You just write a program, use Smith-Waterman to get rid of it. That will be fine. And next step is they want to do the clustered reads, the sequence reads. These are the processed reads into a unique sequence tags, okay? Which means that if this is a 22 base pair mer, and if there's a lot of PCR thing happened on one microRNA, there's got so many different molecules, they are going to combine them, just only use the unique ones at this stage. And the next step is they will match the identified non-coding RNA with existing database, including the RFAM. Okay. RFAM is a collection of RNA families, each represented by multiple sequence alignment, consensus, secondary structures, and covariant models. And this is a database, and it includes many different types of non-coding RNAs. Most of them we are not interested in, but they are sequenced anyways. So this is a kind of, you can consider this as one of the filtering steps. You want to get rid of those. And further, you want to match your stuff with the known microRNAs in the mirror base, okay? And we know that most of microRNA product is already documented in the mirror base. And then you can further make the story out of that. So this is what the DSAP pipeline is doing, okay? But if we use this strategy, we are only focusing on known microRNAs, not unknown ones. But how about unknown ones? We don't want to be wasting those information. And this is a paper published in 2008, very early actually, in the Nature Biotechnology. And they are based on the microRNA biogenesis process. This is for the de novo identification for the microRNAs based on the microRNA sequencing data. So what happens is they are based on this structure. So if this is a microRNA precursor, okay, it will form this type of hairpin loop. And when the dicer cleavage comes in, and this part will be cut off, okay? When you sequence it, and you want to see the sequence reads align in this way. So most of the sequence is stacking in those regions, and this will be the star sequence. If it's not, there's no microRNA there. Some of them do have it. If it's not there, and these two will actually be degraded by the cell. So this is the pattern you want to look at. There's a significant amount of arrays that's stacking around this mature microRNA location, but around this loop, there's not too many, okay? And if this is not a microRNA product, and the non-dicer process or degradation involved, so what you are seeing here is you are really breaking this hairpin loop potentially at any place. And after you do the sequencing, and you are observing that some of are aligned to here, others are there, and sometimes it's connecting those two. We know dicer doesn't work in this way, right? If dicer is working, and this part will be cut. So based on this principle, of course, most people say, before we have this paper, and most people do is they got this enrichment on certain part of genome. They think this is a potentially microRNA site, and they just only focus on the hairpin loop. They say there's a signal there. It can potentially become a hairpin loop. That's a potential microRNA. And I think this paper, using this information, really increased the specificity of the microRNA detection. And their workflow is kind of pretty simple. The first step is they will first filter based on other RNA species, okay? And because we are sequencing on microRNA with data size selection, they are very short, we know that. But other rRNA are pretty short, tRNA are pretty short, and those RNAs that you are not interested in may still be in the final product. And so you want to filter your species based on the existing database. And then next step, you want to do the secondary structure prediction. You want to see whether there's a hairpin loop there, right? And because the microRNA biogenesis, we know hairpin loop has to be there. If you see something like this is not happening, we need to throw that away. That is already remove some information. But most people stop there. And by using the microRNA sequencing, the dicer principle, we are specifically looking for this type of pattern on the mature sites. There's some more reads stacking there. So they designed a probability model based on read distribution, and actually it's log all the probability of a sequence being the real microRNA precursor versus the background hairpin. And there's a software package available. It's called microRNA Deep. All right, I haven't tried, but we are going to try it very soon. Some more information about microRNA. And the question is, in a very early stage, we've already talked about this. This is the assay itself, but is this assay really accurate? So a paper published this year, and they found out sometimes it's not. Right? The reason for that is once you do this, the ligation can cause a serious bias. Ligase has a sequence specificity. Okay? And if you look at this, they sequenced the 3 microRNAs here. They evaluated 3 microRNAs here. And this plot is kind of, so each individual one pair here is this kind of FNN and ENN. So based on different sequence features they are trying in this the process. So you can see that inner circle represents the percentage contribution of each adapter combination to a particular microRNA that was sequenced. One very striking thing here is that there's a green line here, and this line is microRNA 294, and if it pairs with ACCG, okay, and this ACCG adapter alone contribute to about 20% of the detection. And this is purely because of the ligase bias. So they are arguing that you want to do the experiment very carefully, and not only just based on whatever available to you, but also think of the design in more detail. And their solution, I haven't read that paper very carefully. I think their solution is to make a combination of multiple things and into the sequencing. And when they design those different adapters, and that can kind of eliminate this potential bias. Another variation is the microRNA sequences also contain variations. And this is another paper published, I think, in Gala's lab from Institute of Systems Biology in Seattle. And they found that some of our microRNAs frequently have sequence variations. They call this as isomers. Okay? And this is the process of generating these isoforms may not be random. Okay? So they found very specific variations, especially very close to the end of the microRNAs. And the mechanism to generate this is not clear at this stage. And mature microRNAs have a significant end heterogeneity here. This may affect consistency and accuracy of microRNA measurements. So these are the things that we need to keep in mind. And they have done very large-scale analysis, and they have put their results in the database. And I encourage you that if you are studying certain microRNA, I did try last night, and if you do microRNA 21, and they have different tissue types, and they will give you all the potential variations and the percentage of each variation in the final microRNA products. I think that's a very rich resource you want to go to look at. All right, we're 20 minutes early, so we still have the time to do the quiz. Great. So next lecture, the first part will be a guest lecture, and Dr. Eugene Ye from IU Bloomington will talk about next generation sequencing in metagenomics. Okay. And the second part, I will take care of that, which is I use ChIP-seq part of 1, which I will give you the biological theories on ChIP-seq analysis and the published studies and experimental considerations. Okay? And for the students who register the class, stay. We need to take a quiz.